Hello, Dr. Bhandari. Today we'll be going over Chapter 16, Lipid Metabolism, and as always, to start off as just a little joke to go on, to just lighten up the chapter. Every chapter is not always easy, but that's for it is, some are others, but just to lighten up the mood, we have three little jokes that we can go over. So it just basically just pokes fun at metabolism. It just saying that for our intestines, and then the reaction and enzyme, how it's just basically stopping it, the reaction of metabolism. And then yeah, let's continue on. So for now, we're gonna go over lipase overview, and then we have our fats that is being basically burned down, and our lipase is just hanging out. And yeah. So lipase. So what is lipase exactly? So in our little cartoon on our left, our lipase is our little bird, and its beak is what's helping break down tag. And tag is triglycerol, and it also contains fatty acid diglycerol, which is being portrayed in the cracker as it's broken down. And the breakdown occurs in the extracellular and the intracellular. So with extracellular, that occurs in the GI tract, as we can see in the birds in the intestine and also the blood. And the intracellular occurs in the lipid droplet. So a fun way to just remember basically the breakdown location wise, we can see that just overall think of a little bird, and then it's occurring into the intestine, your tag and all the two others, your fatty acid and diglycerol. And as for the intracellular part, you can think of feathers, the bird's feathers being hydrophobic, is if you ever witness out in a park where their little birds are bathing in the little water, you can see that basically the water just falls off from their feathers, being a little hydrophobic. So that's a fun way you can think of that. And for our extracellular, so we're going to go in more depth for our extracellular breakdown of tag of the GI tract. So we first start off with our tag, of course. So this is our dietary triglycerol. So from there, and it's emulsified by our bile acids. So to just have like a good memory of like how this overall structure is, I drew tag as our little jellyfish essentially. So those will be, just for reference, those will be our tag. And then afterwards, once they're there, they will be, so our second step will be lipase cleavage, as we unfortunately see our jellyfish, chopped from the head and the tail, or the little arms. So our top head will be our glycerol, and our little tentacles will be our fatty acids. And from there, it's gone into the intestinal epithelial cell. We'll just put this as an intestine, for better memory. It's just, fun cartoon. And then from there, those little parts that we have mentioned from the glycerol and fatty acid, they go and recombine inside to get our jellyfish back. Good. And then we have our chylomicron. So it's basically entrapped inside. And then our little, what you can say, jelly beans are basically our lipoprotein and our phospholipids. So it's just hanging out in there. And then from there, from our left side, the chylomicron is exported into the lymphatic system, which it can enter into the circulatory system that we have on our right, which is transported into the peripheral tissues. So we have a little nice diagram. Normally, when you're looking at tag, I believe there's just a handful, and there are a few. There's not really much of like a specific number, just, few there, and then unfortunately a little chopping there, but it basically goes into its system and it runs back to our circulatory system to keep us all good. And now for the extracellular breakdown of blood. So again, we have our chylomicron, and then our little jellyfish is our tag, as always. And then as for our jelly beans from last time, this is now APOC 2 that we have indicated as an orange or little beige type of color. And then our green is our ApoB48. And then from there we have little pieces of our glycerol and our fatty acids. And for the APOC2, it is binding with a lipoprotein lipase in the endothelial cell, or we can use again as our intestines. From there, we drew as lipoprotein as a lip, so it'll be better remembering. So, it binds lip, and then it goes, and then we have our fatty acids on there, and then which would enter inside, we can see in our image from here. And not to forget, from the glycerol, we can see that it's going to the liver, which is essentially going to the blood. And then, yeah, and then so from there, our intestines, it goes to formation of albumin fatty acid complex. Our fatty acids basically enter any nearby cell, and we have an egg as an example for the albumin fatty acids. So for me personally, I just remembered that a long time ago that somebody said albumin is usually a egg white, so I just kind of associated with that. So our eggs right there. And then from there, it's transported through a microcapillary, so that's a little truck, to any distant cells. Yeah. So now the intracellular breakdown of TAG and adipocyte and lipid droplets. So again, we have our chylomicron, a little jellyfish, all intact, so we're all good. And our yellow, as we can see in the inside with the jellyfish, are indicated as lipid droplets. And the outer perimeter of that is pepilipin. It's a little tongue twister, but we can think of like a pen, or essentially just a little bracelets going around. And then in the outer side, you can think of really like it's a bowl of Lucky Charms that we have on our side. So we have here our hormone sensitive lipase, our adipose triglyceride lipase, our regulatory protein G58, and monoacylglycerol lipase in our nucleus. And then above, we have a glucagon receptor, which is our light blue. And then we have a B2 adrenergic receptor. That was a mouthful, but basically it's basically what's occurring into the lipid drop in the droplet. So essentially we're still keeping up with just chylomicrons from the previous slides. And from here we have our fate of glycerol release from TAG. So on our left, we can think of glycerol kinase activating glycerol at the expense of ATP. So basically, we have a little funny image of just, money representing the expense. And then the oxidation, it's via oxidation via glycolysis, which is 5% energy of fat that comes from the glycerol moiety. So again, we can associate the 5% energy from the money from the left image. And alternatively, glycerol 3P can be used in TAG, which is our little jellyfish from before, not to forget, just association, or glycophospholipid synthesis. So yeah, it's a little fun image. Just remember, a little jellyfish are tags. And then, of course, we have our fatty acids, which are activated and transported into the mitochondria. And we have our joke, as always, mitochondria is the powerhouse of the cell. And from there, we can see that beta oxidation of fatty acids occurs in the mitochondrial matrix. So from there, the fatty acids are then transported to the mitochondria via the acyl carnate or carotene transporter. And to remember just carotene or carotene, we can think of like carnivore, so meat. So I have a image of basically a nice juicy steak that we would all love to have. And from here, the fatty acid involves the conversion of fatty acid into fatty acyl CoA and the formation of fatty acyl uh, carotene, and then transfer of fatty acid into intramitochondrial coenzyme A. And for the reason why I have a col is to represent throughout the slides a coenzyme A. So just remember like co is col. So yeah. And then coenzyme A. So or CoA for short, and this is derived from vitamin B5, and from many of the groups avocado is part of the vitamin B5 group. And then from here we have a more in-depth structure of what coenzyme A is essentially. So on our left side we have our reactive dialkyl group, which is what's forming that dialkyl ester that we see in coenzyme A, which is the high standard free energy of hydrolysis. And then our middle part is our pantothenic acid residue. So we have that going, and then we've pretty much seen this nice looking structure in our nucleotide, our DNA, RNA. We have our phosphorus, we have our phospho group, our ribose, phosphor group, all the good stuff. And this is essential micronutrient. Remember, essential is what we grab or we get from other things to aid us. So it's not basically naturally produced in our bodies. And remember, coenzyme A, col, and yeah, we're all good to go what coenzyme A is. And a little bit more just in depth for the conversion of fatty acid to fatty acyl coenzyme A that we just labeled as FA fatty acyl coenzyme A. Excuse me, that's a huge tongue tie. And then of course it's gonna be occurring into the side of the mitochondria, of course, powerhouse of the cell. So our fatty acyl coenzyme A synthase catalyze formation of the fatty acyl coenzyme using the ATP coupled mechanism of the systolic side of the mitochondria. So we're gonna go over the two steps. So in our fun little image, as seen previously in the other one, how we have drawn our phospho group, we have our three little people. So that represents our phospholipid group essentially, and it's connected with a adenosine, or we can think of as like an advertisement for short. Just so we have three connected to an ad, so an adenosine or an A, and from there in our right image we see that our third little um, phospho group is being now grouped together to a fatty acyl CoA. So this is a CoA, coenzyme A, and then we have our little phospho, and then of course it's still with our adenosine, which is now our AMP, as seen here. So its intermediate is attacked by the thiol group of coenzyme A, forming that fatty acyl coenzyme A, releasing that AMP that we need. And then from there we can see that our fatty acid essentially left our little phospho group or little person, and then it's seen with the combination of coenzyme A. And again, our coenzyme A is represented as our col. And then on our left side, we can see that our two little buddies formed there, and it's rapidly hydrolyzed to 2-pi by enzyme of pyrophosphate. So we can see that we can associate that these two just went out and then just grabbed two little pies, or excuse me, two huge pies for themselves, while their friend, who knows where, went with the fatty acid. And then our fatty acid, our beta fatty acid essentially, is the fatty acid coenzyme ester formed at the systolic side outer mitochondrial membrane that we've mentioned before. And this can either be transported in the mitochondrion for oxidation or used for systole to synthesize tag, as seen here. And then one destined for oxidation are transiently attached to the OH group, as we can see in the previous slide of the many OHs we've seen, and group of cardenine for form of fatty acyl cardenine. And right here we can see that there's a little example of our cardenine. And yeah, thank you for staying. I hope we make it a lot more simpler.